Today the topic of our discussion is ABO and RH blood grouping system, and along with that we will also learn how to determine the blood group of a person. What is ABO blood grouping? So ABO blood grouping, this system is based upon the absence or presence of a specific inherent antigen on the surface of the red blood cells. Now we have four types of blood group: blood group A, B, blood group AB, and blood group O. Now these groups are determined by the presence and absence of the antigen on the surface of RBC. So for blood group A, we have antigen A presented on the surface of the RBC. So it is represented here by the pink circles. For blood group B, we have the antigen B presented on the surface of the RBC. And for blood group AB, we have both antigen A and antigen B on the surface of the RBC. And for blood group O, we have none of the antigens that are present on the surface of the RBC. Now, since the antigens are present, now corresponding antibodies will also be present. So for blood group A, we have anti-B, that means antibody for B. Now see, a person having blood group of A has antigen A and they have antibody of B. That means in case the blood group of A, that blood is mixed with the blood having the blood group of B, then what will happen is that the antibodies that are present in blood group A, that means the antibody B, that means the antibodies for antigen B. So these antibodies will bind with the antigen of the blood group B and the agglutination reaction will occur. So that means these two bloods are not having a match. So this is the differentiation. Now in blood group B, we have antibody for antigen A, that is anti-A, for the same reason. Now for blood group AB, we have none of the antibodies. It is very easy to understand because both the antigens are present here. Both antigen are present, that's why no antibodies are present. And for the blood group of O, we have both the antibodies. Because you see in blood group O, we have none of the antigens. No antigens are present, not A, not B. So the antibodies, that means both the antibodies are present. That's why we have no antigen present on the surface of the blood group O. Now this A and B, these antigens, these are actually oligosaccharide chain. These are different oligosaccharide chain present on the surface of the RBC. And the antibodies, these antibodies present in the plasma, they are IgM, immunoglobulin M. It is because you see these are pentamers. Each of these antibodies are pentamers. That means there are 5 immunoglobulin complex in one. So these are IgM. Now we will talk about the biosynthesis of the antigen A and antigen B. And for this, we will consider this reaction. So for a person having a blood group of O, they have H antigen in them. But H antigen is present in both blood group A and B. Now the differentiation between the antigen A and antigen B depends on an added compound that is present with the H antigen. Now this biosynthesis depend on the presence of enzyme, different enzyme in the person. So there are two types of enzyme, GTA and GTB. If a person has GTA, that means 3-alpha N-acetyl galactosamynyl transferase enzyme, if a person has this enzyme, then they will utilize the UDP GalNAc substrate, that is uridine diphosphate N-acetyl galactosamine. This compound is used as a substrate for this GTA enzyme, and what will happen is that they will transfer this GalNAc compound to the H antigen. and as a result the A antigen is formed, and so our blood type is now A. If a person has GTB, this GTB utilize the UDP gal substrate, that is uridine diphosphate galactose substrate, and they will transfer the galactose from UDP gal to the H antigen, and as a result we have now the blood group of B. Next we will talk about RH blood grouping. Now this RH blood grouping, they have also antigens. So these RH antigens, these are some protein complex that are present on the RBC surface. The genes RHD and RHCE code for RHD and RHCE proteins, and RHD, they carry the D antigen, and RHCE carry these 4 antigens, that is capital C, small c, capital E and small e antigens. So we all have this RHAG, this protein in our body. Now this RHAG protein, they helps to transfer the RH antigens to the surface of the RBC membrane, and in case this RH antigen, that means the D, C, E, these antigens, if these antigens are not present and not expressed, then the integrity of the RBC will not be there. So the important role of this RH antigen, make sure to remember that RH antigens play a role in maintaining the integrity of the RBC membrane. Now what I just told earlier, here it is a representation. So the RH positive, when you call a person RH positive, it means they have both the genes, that is RHD, RHCE, and both since they're both present, so all the antigens are also coded by this gene. So RHD, it will code for the D antigen, and RHCE will code for the C and E antigens. When a person is RH negative, it means they don't have the RHD and no D antigen is coded by them. So the differentiation between RH negative and RH positive is only depends upon the presence of D antigen. Now here, this is the representation of the proteins. Now this protein, actually it is a transmembrane protein, and it traverses the cell membrane of the RBC 12 times. If you count these loops, you can see there are 12 times the protein crosses the surface of the cell membrane of RBC. And another picture is also there for RHCE. Here you can see that at the top it is written C/small c. Now this antigen, there are 4 types of antigen that can be coded by RHCE, and the differentiation is given here. So if we have capital C antigen, it means that at the position of 103 we have serine, and if we code for the small c antigen, then at 103 we have proline. And similar to that, if we code for the antigen E, that is capital E, then at 226 position we have proline, and for small e antigen we have alanine. Now we will talk about the laboratory technique to determine the blood group of a person. So these are the equipments that we need. So we need a clean grease-free slide. So this is the kind of slide that we use, having 3 dents on the surface, and the first circle is called the A, second is B, and the third one is D. Remember this part. Next we need cotton, spirit, needle, sterile sticks, and antiserum ABD. Now the procedure. So you at first make sure to clean your fingertip with the alcohol, and then prick your finger with the needle. Make sure everything you do, it is in a sterile condition, and you will put 1 drop each on these 3 circles. Next, you take those antiserums and you put antiserum A in the first circle, that I told earlier as circle A, and next add antiserum B in the second circle, that is denoted by B, and in the D circle add anti-D. Now mix these reagents with the blood drop and you will get results something like this. Now it's like for a person. One example I am showing here is for a person, and you see that the agglutination occurred in second and third circle. Now this indicates that the blood group is B positive. Now why is that? So when we talk about antiserum A, now antiserum A, this reagent has antibody for antigen A, and when we consider this person's blood is B positive. That means they have the RBC that represents the antigen B on its surface. And so when we add the antibodies for A antigen, obviously the reaction will not occur because there is no A antigen present. On the other hand, when we add the antiserum B on this same blood, we see the agglutination because antiserum B has the antibodies for B antigen. So they will come and bind to these antigens, and thus the agglutination will occur. And for anti-D, that means it will determine the presence and absence of the antigen D. So when you add this, it has antibodies for antigen D. If antigen D is present, then it will coagulate, and if it is not, then the blood will remain normal. So you see the coagulation is in the B and the D circles. So as a result, we can say that the blood group is B positive. So these are the reasons that I already discussed. You can read it from here. And here are all the possibilities that can occur. So I will discuss few of them. So if we consider A positive, so A positive means the blood group has A antigen present, and if A antigen is present, the antiserum A reagent will bind to the A antigen, and as a result the agglutination will occur. And since it is A positive, that means the antigen D is also present. Since D is present, the anti-D will bind to the D antigen, and as a result the agglutination will occur. If we consider the AB positive, now in AB positive all the antigens are present, antigen A, antigen B and antigen D. As a result, all the antiserums will bind to the specific antigens and the agglutination is seen. In case of O negative, in O negative none of the antigens are present, because antigen, you already know that O means no antigen present, A or B, and it is negative. Negative means no D antigen. As a result of this, we will get no agglutination reaction occurring in none of the circles. As a result, we can determine that is O negative. And in case if the O blood group has a O positive type, then this positive means D antigen present. D antigen present means the D antigen will bind to the antiserum D. This way we can determine the various types of blood groups depending upon the agglutination reaction. Thank you for watching and subscribe for more updates.